Okay, on with research methods. Research methods lesson 3, control demand characteristics, investigator effects. Not as much today because I think some of these are quite tricky. So I'd rather we pause, have a good look at these, understand them well, rather than piling on more. We can do more in the subsequent weeks. Okay, so just a small amount that I want you to know well. Here's a specification. I won't read it through, you've got it there for you, but a lot of terminology that goes for research methods as a whole. There's got to be terms that you can define straight away without hesitation. We will do a lot of work on that when we hopefully are back together next year. So as we've been doing this alongside the research methods topic companion, so it's pages 38 to 40 for today, whether you have that open alongside this or you look at it before or after, whatever, that's what we're referring to today. So the first one is extraneous and confounding variables, sometimes sort of used interchangeably. We need to be a bit more careful with that. We need to separate them. I've had a look into this. This is a tricky one. Teachers often asking me what's the difference, or to clarify, not so much that, but how do you get the difference across the students. So I looked into this again today, and the consensus seems to be don't overcomplicate it, keep it simple. So extraneous variables, anything other than the independent variable that might affect your dependent variable. So going back to what we talked about last time, in an experiment you want everything to be constant between the conditions. Only thing that should be changing is your independent variable. If the dependent variable changes, you can say that's due to the independent variable. Now we're saying extraneous variables are anything that might potentially, emphasis on the might potentially, affect the dependent variable. Now confounding variable is when one of these has affected the dependent variable. Okay, that's as far as I'm going to go with this. If you want to know more on that distinction, then do read up from the usual online resources or the textbooks yourself to get a bit more depth to it. But actually, broadly speaking, it's not worth the hassle. I'd rather have you all knowing this distinction clearly. So we've got that at your disposal for a definition or to identify these from a scenario. But if you want to know a bit more, then yes, of course, read into it a bit more. But as you can see from the diagram here, at the top we've got what we want to happen, that the independent variable is changing the dependent variable. But these extraneous variables, if they do affect it, then that would be classed as a confounding variable. Now we use the word control a lot, particularly in experiments, and there's lots of things we can do to try to control these potential extraneous variables. So things like random allocation, counterbalancing, randomization, standardization. So random allocation, this is important. Participants should be randomly allocated to the conditions. So particularly if you're doing an independent groups design, then it should be random allocation to condition A or condition B. This, as we said last time, reduces the risk of participant variables in the form of individual differences that could affect the results. So if I did, let's say I had a year 12 class, let's say that, I know it's not, but if my classroom was split equally down the middle, I said, okay, students in the left, you're doing condition A, students on the right, you're doing condition B, then there's a chance that those aren't equivalent. There might be a difference between one group of students and the other. It might be the studious types sit on the left, the less studious types sit on the right. I usually go for the front of the class, back of the class, but that's just reinforcing stereotypes. We wouldn't have an issue with the back rows at all. So we'll go with the left-right distinction here. So that would be quite sloppy. We shouldn't do that. So what I'd do is I'd take all of those names, and then using similar processes to random sampling, which we'll talk about when we do sampling, be it named out the number allocation, anything, we would randomly assign those pupils. That should help to overcome the risk of these participant variables. Counterbalancing, we did this last time quite a bit, so I'm not going to hang around on this. Make sure you're using these terms appropriately. That's one of the main things they want to look at with research methods. It's not it's not a great depth, it's just that you get it. You know how to apply these and you really understand them. So if you misuse this, if you say, oh, independent measures counterbalancing, they clearly, you don't know what you're talking about. So please make sure you're clear. Counterbalancing is for repeated measures design and overcomes the risk of order effects. And remember, it doesn't get rid of or eliminate order effects. There's just as many orders to what's being done here, but it balances them out, nullifies them, cancels out. Use that kind of language. Now, randomization. This word, I was looking into this as well today. Sometimes this seems to be used interchangeably with random allocation, but they are put separately on specifications. So I would keep random allocation as one thing, and then for randomization, I want to focus on what's actually being used within the study. So the stimuli, etc., that they're being presented. So if you had a memory task like the ones we did at the start of the course, I was presenting words in an order to everybody. Now, if we were doing that properly, first of all, it wouldn't be done to a group, it'd be done individually. And rather than always having those words in the same order, they would be randomized so that each participant sees them, the same words ultimately, but in random order, so mixed up differently, just in case there's any impact of the order of the words. So it might be that some of them form associations more readily than others, and that could influence your findings. So if they're randomized, that would be cancelled out. So randomization is what's actually being used within the study, whereas random allocation is the participants before you even start doing the study. Okay, so please get the distinction between the two. Standardization, standard procedures. We want, as we've said multiple times, everything to be consistent with only the independent variable changing between conditions. So the environment, the conditions in which the study is taking place. Don't get too carried away with this. I have had people going into quite obscure external factors that really wouldn't be an issue. But within the lab setting, or whatever setting you're in, keeping it as constant as possible. So same place, same equipment, same materials. Things like if there's noise in one condition but not in another, that's a big potential extraneous variable there. The lighting, the temperature within the room needs to be consistent. Things that could realistically affect the study. So just don't go too wild, don't get too tenuous with your ideas here. There should be fairly obvious things that you can think need to be consistent. Time of day, that's important. I certainly know that I'm a lot more efficient in the mornings than I am post lunch. So people might be one way or the other. Better to keep that consistent. The time of day you're doing the study. And a crucial one is standardized instructions, so that all participants receive exactly the same instructions. I can deliver, in theory, the same lesson 3 times with you, the 3 different groups. Doesn't mean that I do it as clearly each time. It might be that I do better after the first one. It might be that I just by chance don't get it across so clearly on another occasion. So standardized instructions, a lot of the times when I did studies, I was asked to read the instructions there, or they're read to the participants. Either way, everybody's receiving exactly the same instructions. Also would be important if you had different researchers. One person could explain it much better than another. If they're both reading in the same script, then that takes it out of the equation. Having these standardized procedures also helps with the replicability side of things. So all research should be replicable by other people. By having standardized instructions and procedures, that makes it all the more easier to replicate the study. Demand characteristics is cropped up last time. We said with repeated measures, there is a risk, particularly with repeated measures, that people can guess the aim of a study, but even with independent measures, people might still think they know what you're looking to do, and they might change their behaviour because of that. If they're not behaving naturally, then that greatly reduces the validity of your study. We need people to be doing what they would normally do rather than what they think they should be doing. So yeah, as I said last time, this can go 2 ways. taking part in studies as a psychology student, yeah, a lot of the time you've got a pretty good idea of what they're doing. You're sort of torn between thinking, oh, let's go with it, and let's give them the results they're expecting. But there is also what's called the screw you effect, which is where you go out of your way to do the opposite. Either way, if you're not behaving naturally, this is demand characteristics, and that is really reducing the validity of what you're doing. So what can you do about demand characteristics? Well, the main thing is trying to stop them from being able to guess the aim of the study. So single blind, a good term for you to use here. This is when only the researcher knows the true aim of the study, that the participant doesn't. Well, you might think, well, fair enough, don't tell them. Now that's an issue because for ethics, we've got informed consent. Participants should be given their informed consent. Now, if you at best haven't told them the aim of the study, or maybe you've lied about it, used deception, then that is actually a bit of an ethical issue. I'm not saying you can't do it, but ultimately it is supposed to be avoided as much as possible. But for many studies to have validity, there has to be something held back from the participants. So single blind, the researcher knows the true aim, they know what the IV is and what's being measured, but the participant doesn't, and there may well have been some deception used. So Milgram, we talk about ethics of Milgram's quite a bit. Ultimately, they were given a false aim. It was, you can see the advert, it was sold as a study on memory when it wasn't. It was a study on obedience. But if you'd said that, people would have known what was going on. Okay, so deception is acceptable as long as it's justifiable. Now, investigator effects, this is when the researcher acts in a way that does support their prediction. It could be conscious. I mean, it could be that you just go out of your way to fix it to get the results that you want. That's quite unlikely, you'd hope. It's more likely to be the more subtle, unconscious cues and prompts that people can give off. You've maybe have experienced this. I ask a question in class, you start giving your answer. I haven't said anything, but you're probably, as you're talking to me, this is wrong. Probably because I might be giving off subtle facial cues and things like that. In comparison to the beaming joy when somebody's saying the right thing. Now, if that's happening, you might be nudging participants towards the answers that you want, even if you don't realise that you're doing it. So how do we overcome this? Double blind. We had single blind before, where the participant doesn't know the true aims. Double blind, neither does the researcher present at the time of the study. So you've got a participant, you've got somebody overseeing the study at that time in that place, but they don't need to know the true aim and nature of the study. They just need to know what needs to be done at that time, what is required of the participants, what they need to say, what they need to measure, all of that. Now, if that person doesn't know the true aim of the study, if they don't know the true independent variable in all of this, then that should greatly reduce the risk of investigator effects as well. You'll hear a lot in the news about medical trials at the moment, and you'll hear the phrase double blind a lot, and that means that the participant, if we say with drug trials, the participant doesn't know if they're being given an active real drug or the placebo, the dummy pill. They don't know that, but then in double blind, neither does the person administering it and monitoring the person. They don't know it either. Okay, so hopefully you can see from that example how this gets rid of all sorts of possibilities when it comes to these sources of bias. Okay, so like I said, there's not very much, but I really want you to see the additional reading as much as part of the task as the questions. The questions are easy. It almost does them for you. I'm going to be reading the same thing over and over, but that's fine. It reinforces what you've been learning. But I think these are quite tricky. So, as I said before, don't just do the bare minimum to get this done. You could get it done in a couple of minutes if you do that. But I need to know And you need to be able to ask yourself, do I get this? Do I understand these terms? So yes, reading the topic companion pages. I'm going to say it is a requirement that you read the digital textbook as well. Just a double page spread, 168 to 169, gives you a bit more depth on all of these as well. Once you've done that, and when you're happy with all of these terms, then yes, get the questions done. But please don't leave anything hanging. Don't have anything you think, don't know what that was all about. That's hopeless. We can't have that. We've got to have you taking responsibility for that. Reading, trying the usual websites. But yeah, email me if there's a particular bit you don't get. I'm more than happy to get back to you. Hopefully some of you have been asking me questions. I'm able to look on a fairly routine basis at my emails, and I can get back to you fairly quickly to help you. But please take responsibility for this. Don't just think, oh well, whatever, the task is done, he's happy, that will do. Okay, I need you to know this stuff well. So those questions, same as before, I've given you the sheet that you need to complete and send it back to me, and that is due on Friday the 26th. Okay, thank you.